Good morning, everybody. Today's topic is epigenetics and early metabolic programming. So, today we will discuss about what we mean by the epigenetics and the consequences of the epigenetics, and also we will give emphasis on early metabolic programming. Now, we are going through the agenda of today's presentation. The agendas are importance of nutrition in early infancy, proteins, the main building blocks of the body, breast milk, the gold standard of protein quality and long-term health, evolution of protein quality and quantity in infant formula, growth and development, clinical evidence with higher quality protein composition. So, what's the importance of the nutrition in early infancy? Disposition to diseases in adulthood are determined by genetic, environmental, and epigenetic factor. So I want to give emphasis on these few lines. Disposition to diseases in adulthood are determined by genetic, environmental, and epigenetic factor. So nowadays there is huge hue and cry about the epigenetics. So what we mean by epigenetics? The epigenetics are the changes in gene function. These are the changes in gene function without affecting the genetic code. What do we mean by genetic code? Genetic code is the genetic sequence. So, epigenetic process brings about changes in the gene function without affecting the main genetic structure, that is, without affecting the DNA sequence. What epigenetic causes? Epigenetic causes aberrant methylation, that means under or over methylation of the genetic sequence. Either a methyl group is added or a methyl group is subtracted from the DNA sequence. But the main DNA code is not changed. This is the main difference between epigenetics and mutation. In mutation, there is changes in the genetic code, and epigenetic processes are reversible, but mutation is irreversible. So we all know about the famous theory of Barker hypothesis. What do we mean by the Barker hypothesis? That is the fetal origin of childhood and adult disease. So there are many diseases which later expresses in childhood or adulthood. They have origin during the fetal period. So epigenetic process plays a role in that fetal origin of childhood or adult diseases. What are the types of epigenetics? Epigenetics are three types. There are nutri-epigenetics, environmental epigenetics, and disease-related epigenetics. So three types of epigenetics. And the word epigenetics was first coined by a British biologist, Conrad Waddington, in 1940. So epigenetic process, it acts like if we compare the gene with a bulb, and epigenetic process is the switch. So epigenetic process controls the expression of the gene. By, as I said earlier, the epigenetic process is a switch. So by controlling the switch, we can control how much light will be expressed from the bulb. So epigenetic process in that way controls how much gene will be expressed and how much gene will not be expressed. That is the switch on and switch off mechanism. This entire process of switch on and switch off mechanism is called epigenetic slicing. And this epigenetic slicing decides many fetal origin of childhood and adult disease. So basic thing is that epigenetic process, it works in early fetal life and during embryogenesis. So early metabolic programming, our objectives today of early metabolic programming. So this early metabolic programming is directly related with the epigenetic process. So we already known about the types of epigenetics. That there is nutri-epigenetics, that means nutrition, epigenetic process, and health. So epigenetic process serves as a bridge between nutrition and health. So many are the diseases which are related with the nutrition, depends on the epigenetic process. In this case, I want to cite a study. There is a study done by a Swedish scientist. He showed us that if fathers are given high calorie diet at their critical period of development, again said, if fathers are given high calorie diet at their critical period of development, it is found that their children are more likely to suffer from some diseases like cardiovascular disease, obesity, and also weight gain. We all know that this critical period of development is around puberty. So this is entirely related with the nutrition, and this is the nutrition and epigenetics interlink we can found here. Another example of this nutrition and epigenetics, that nutri-epigenetics, is there is a famine occurred in Netherlands in around 1944-45, and during that famine period, the entire population of Netherlands is deprived from huge calorie deficiency. The general calorie intake of the entire population become less than 500 kilocalorie. So the children in utero, they also suffer from severe malnutrition or calorie deprivation. And later on, it is found that because in utero there is lack of calorie because of famine, the fetus in utero, they are programmed to conserve energy. They are programmed to conserve nutrients. And this is the epigenetic process came into role at this stage. And those child after born, if they are exposed to high calorie diet, they are more prone to develop obesity, because in the fetal period they are programmed to conserve energy. So when they are exposed to high calorie diet after birth, that is early fetal programming comes into play, and they try to conserve more calorie, and there is a more chance of become obesity and obesity related complications. So these are the few examples we can give relating with the nutrition and epigenetics. So the second type of epigenetics is the environmental epigenetics. There are many environmental factors which can influence the epigenetic process. So what are the environmental factors? We can give example like smoking, pollution, hormones, there are some diets which can also influence the epigenetic process. So environmental factors plays an important role in influencing the epigenetic process as early in fetal life and early embryogenesis. So early nutritional environment interacts with an individual's genetic disposition to program metabolism and development. So relation with the nutritional environment and epigenetic process. The early epigenetic determination of constitutive gene expression is likely to be one of the keys to understand early life programming of metabolic disease and obesity. There is a figure, we can see how epigenetic regulation can occur. There is some study. One study shows 653 subjects, 32 of them were overweight adults. The study result shows weight gain during the first week of life and during the first 112 days of life was associated with adulthood overweight status. Another study shows the subjects were 216 premature, 61 control, FMD at 13 to 16 years of age. Result shows greater weight gain or linear growth in the first 2 weeks postnatally was associated with lower FMD at adolescents. FMD was 4% lower in adolescents, with the highest compared with the lowest rate of weight gain in the first 2 weeks of life. There is a very interesting slide which shows the relation with breastfeeding and later obesity. We all know the importance of breastfeeding, the exclusive breastfeeding, its positive effect on newborn and infant, also breastfeeding effect on maternal health. So this slide shows a cross-sectional survey in Bavaria, Germany. 134,577 children at the time of entry to school. In subsample analysis, children who were never breastfed had a higher prevalence of overweight and obesity than the breastfed babies. Longer duration of breastfeeding was associated with a lower prevalence of later obesity. So in relation with this slide, I like to give emphasis on pregnancy and epigenetic process, because all we know that I said earlier, the epigenetic process begins in fetal life and embryogenesis. So there is a critical period of pregnancy while epigenetic process plays a very important role, and that critical period of pregnancy is the first week around conception. So maternal diet, maternal mental and physical abuse during pregnancy, maternal disease, all has influence on the epigenetic process, and this influence, can switch on or switch off the epigenetic process in such a way that will affect later fetal development, and also has implication of later childhood and adult diseases. So maternal environment during the first weeks, or 1 or 2 weeks around conception, is very important. There is some study which shows that if the mothers are abused during pregnancy, physical or mental abuse, there is an changes in the epigenetic process, and which affect the hypothalamic, pituitary and adrenal axis. And this epigenetic effect, this aberrant effect on the hypothalamic, pituitary and adrenal axis of this fetal, later on these babies, when they're born in childhood, they have many behavioral problem, aggressive behavior they shows, and also mental instability. So let's come to the main slides now. The overweight infants have a 4.14 higher odds of childhood overweight compared with those who were not overweight, independent of feeding time. Exclusive breastfeeding did not affect the association between infant overweight and childhood overweight. Our main topics is the protein quality for long-term health. So before going into that another domain of protein quality, I'd like to give some emphasis on other few aspects of the epigenesis. So we already discussed about the main types of epigenetics, that is nutriepigenetics, also the environmental epigenetics, and lastly I want to give emphasis on disease-related epigenetics. So there are many diseases which we can relate with the epigenetic process. The first disease related with the epigenetic process is the colorectal cancer. The study shows that colorectal cancer cells has aberrant DNA methylation. So that means the cells which turn into colorectal cancer cells, they have less expression of some genes, and this is due to aberrant methylation, that is either under or over methylation of the DNA, that is related with the epigenetic process. There are some few examples of other diseases which we can relate with the epigenetic process. Those diseases are Fragile X syndrome, and very importantly, another domain is very widely discussed nowadays, autism. And we can relate autism with the epigenetic process. Epigenetic process in autism babies, it was found that there is aberrant methylation of the oxytocin receptor. So we all know that oxytocin receptor are related with the behavioral pattern. So in autistic child, the scientists found there is aberrant methylation of oxytocin receptor. So this way we can relate many diseases with the epigenetic process. Our next agenda is human milk, the gold standard for protein quality. Till now we are discussing about the early metabolic programming, the fetal origin of the childhood and adult diseases, and epigenetic process. So now we switch on our topics to protein quality and relation with breast milk, human milk with protein quality. We all know the composition of human milk, and in the slide which we are showing the different composition of the human milk and cow's milk. So in human milk, whey protein is 60%, in cow's milk it is 20%, casein in human milk is 40%, casein in cow's milk is 60%. We all know that. And there is another slide showing other composition and difference between the human and cow's milk. The slide showing that alpha-lactalbumin is more in human milk, beta-lactalbumin around 40% in the cow's milk, immunoglobulin is around 15% in the human milk, as well as 95% immunoglobulin A. Lactoferrin is more in human milk and trace amount in cow's milk. This is an interesting slide. This is showing the variation of the human milk content as the newborn baby grows older. So first the colostrum, then the transitional milk, and then the mature milk. So the slide shows in the colostrum the fat content is around 21 gram per liter, but you see In the mature milk, it is about 40 gram per liter, that almost double. The fat content almost double from colostrum to mature milk. And the protein content, on the other hand, it decreases. In the colostrum, the protein content is the highest, 31 gram per liter. Then in transitional milk, it is 9 gram per liter, and in mature milk, it is 8 gram per liter. And sodium content also decreases as the milk becomes more mature. We all know that there are some bioactive peptides human milk contain, the breast milk contain, which has many advantages. Physiological activities provided by the milk proteins, mainly in the gastrointestinal tract, GI tract, those are enhancement of the nutrient absorption. This bioactive peptides enhances the nutrient absorption. They inhibit some of the enzymes. Some bioactive product has beneficial enzymatic activities. They stimulate the growth. They also modulate the immune system and provide defense against the many pathogens. Human milk also contains some hormones. For example, the insulin, cortisol, thyroxine, insulin-like growth factor, GIP, VIP, somatostatin, estrogen. There are many hormones. This slide shows the insulin concentration. Insulin concentration of the human milk is significantly higher than the fresh pooled cow's milk. Insulin is barely detected in the infant formula. This slide shows the insulin content in human milk after 1 year of breastfeeding. The average insulin content is around 42.6 microunit per ml. This is a slide showing percentage of essential amino acids in human milk versus bovine milk. We can see tryptophan content in human milk is higher than the bovine milk. In bovine milk, the tryptophan content is low. Leucine content in bovine milk is low. On the other hand, the methionine, lysine, and histidine in the bovine milk is much higher than the human milk. Human milk, we all know that, is a high quality source of protein. So we are actually nowadays giving emphasis on the quality. If we can give higher quality protein, we can compromise on the quantity. So this slide shows the human milk. The quality of the human protein in human milk is much higher than the bovine milk. You see the tryptophan content in the cow's milk or bovine milk is much less than the breast milk. Protein quantity in human milk declines rapidly within the first few weeks of life. The quantity declines, but the quality of the protein in human milk is much higher than the cow's milk. The slide shows we can see in the earlier slide it also shows that as the milk become more mature, the protein content, quantity of the protein gradually decreases. So the colostrum, transitional milk, mature milk, the slide shows the protein content gradually decreases. High quality protein in human milk allows low protein quantity, which I already said. If we can provide high quality protein, we can little bit liberal on the protein quantity. According to the WHO, FAO, UNU expert opinion, the human milk content of amino acid is the best estimate of infant amino acid requirement. A low protein quantity is possible only if protein quality is high, that is, quantity of individual amino acids and ratio between the different amino acids. The primary limiting factor in reducing the total protein concentration in infant formula is the ability to provide sufficient quantities of essential amino acids. In these slides, the difference between the composition of human milk and cow's milk, we are giving importance in this slide on GMP, Glycomacropeptide. You see in this slide, the GMP content of the cow's milk is around 12% and is almost nil in human milk, the GMP content. So what is GMP? Glycomacropeptide, present in whey as a byproduct of the cheese making process. The function of the GMP, inhibitory effect on acid gastric secretions, modified blood concentration of digestive peptides, immunosuppressive and immunostimulatory properties, induced satiety, not proved in human model but in animal model, but the effect not seen in human studies. Evolving from conventional to the gold standard. So we are talking about always the higher protein quality. Addition of beneficial proteins, that means the alpha-lactalbumin, bioactive peptides. We already know that bioactive peptide has many beneficial effects on the GI tract, like immunomodulatory effect, anti-inflammatory effect, So removal of unnecessary cow's milk proteins, beta-lactalbumin and GMP is necessary. So are we heading for lower protein content? Probably, yes. So this slide shows the protein concentration. We all know that in human milk is different from cow's milk. So protein content in the infant formula. The slide shows in 1970s the protein content in infant formula is around 4 gram per 100 kilocalorie. In 1980s it decreases to 3 gram per 100 kilocalorie, and at present it is about 2 gram per 100 kilocalories. So in infant formulas over the years the protein content is gradually decreasing. The aim is to match the protein content of the breast milk. So in this slide on the left hand side shows that the protein content of the cow's milk is almost 3 times than the human milk. So our aim is to match the protein content of for infant formula close to human milk or breast milk. The global standard for macronutrients composition of an infant formula, we are targeting the protein content, as I earlier said, close to human milk. So our target is the content of protein in infant formula will be close to 1.8 gram per 100 kilocalorie. The panel considers the EPSA opinion. Based on the available evidence, a minimum protein intake of 1.8 gram per 100 kilocalorie from infant formula based on intake milk protein is sufficient to ensure adequate growth and development. So, if we can ensure the protein content or protein quantity 1.8 gram per 100 kilocalorie infant formula, that is sufficient to ensure adequate growth and development. Minimum amount should be understood as target values which cover the nutritional needs of virtually all infants born at term for optimal growth and development. Nutrients which are not used or stored have to be excreted, and this may put a burden on the infant metabolism. So the question is minimum and what is optimal. We are almost end of our presentation. There are a few slides left. This slide shows a study done on different infant formulas and follower formulas. They show the protein content varies from lower protein to higher protein. Ranges in the infant formulas 1.77 protein per 100 kilocalorie, and the higher group is 2.9 gram per 100 calories. In the follower formulas, the range varies from 2.2 gram per 100 kilocalorie to 4.4 gram per 100 kilocalories. There is a wide range of difference between the protein content. This slide shows the lower protein group had comparable BMI to the breastfeeding group at 6 years of age. The comparison in the graph. Protein content of 1.6 gram per 100 kilocalorie with glycomacropeptide removed, mothers of infants in breastfeed group had significantly lower BMI. So, in conclusion, we can say human milk is the gold standard for protein quality and quantity. Bovine milk protein composition differs significantly from human milk. Protein quality in infant formula has evolved over the years and should be as close to human milk as possible. As we said earlier, 1.8 gram per 100 kilocalorie. Improved protein quality closer to that of human milk may have positive impact on long-term metabolic health. Thank you.